Hello, good afternoon, everyone. It's been a long while since my last class. I hope that you still remember me. Okay, if not, it's fine. I think I might have some new viewers. Okay, it's fine. I'm just making now finally that I've done my work that is been bothering me, so that now I have the time to make some more classes. Okay, since the exam season is quite near, so my focus nowadays is to make questions, is to discuss questions that are challenging. So my focus for this year is to do some challenging questions, which students may find difficult to answer. Okay, some of it may be easy to you, all right, but might be difficult for others. So it might be easy for you, but it might be difficult for others. All right. So today my topic here is, I think you will know when I share my slide. So today my topic here is a Form 5 syllabus, Form 5 chapter, which most students will find it difficult. Okay, so I'm sharing my slide here. All right, so what am I doing today? All right, so today's class is in English. I will explain in English. But on Monday, I'm going to have another class with the same topic, same slides, but I'm going to explain in DM. So my slides, don't worry, because they're in Bahasa, that means in double language. You will see the answer in English and also in DM. Okay, all right. Now today, I find many students are finding it difficult to answer questions related to genetic engineering. Okay, this is the topic which quite a lot of students do not get. All right, and so I've dedicated today's class using 3 questions. I'm going to discuss 3 questions which are related to genetic engineering. All right, let's start now. Yeah. Okay, if you'd like to let me know who you are, you can just type hello and I will see your name there. All right, if not, so far at the moment, I do not know who's with me, but it's fine. All right, just follow the lesson. Okay, now today, question here. Now this question, most people will find it easy. All right, so this is the basic. Now this diagram shows a pentose sugar molecule. So they give you a pentose diagram here, telling you that you're supposed to complete the diagram. All right, now draw and label the diagram to complete a nucleotide structure. Okay, so you need to remember the basic parts of a nucleotide molecule. What is shown here is the pentose sugar. So you have another 2 more, we call it 2 more portions, 2 more parts, which you should know how to add in and label. Whatever you do, do not forget to label, because if you do not label, you will not get your marks. Okay, you must remember to label. You have to label. Okay, so this one here, okay, this is the phosphate group. Okay, ataupun kumpulan phosphate. All right, okay. Then the other one on the other side, you must label at the correct place. Yeah. The other one here is the base for nitrogen, or we call it nitrogenous base. Okay, so this question, 2 marks. So 1 for each. 1 for kumpulan phosphate or phosphate group. The other one is nitrogenous base. Make sure they are labeled correctly at the correct place. You get 2 marks. Okay, so this is, I think, very basic and it's easy for some people, most people. Okay, let's go on to question B. If you have any questions, you can put in your comment in the comment here, right? And then I'll read your answer. You'll read your question. All right. Okay. Next. Now, what about the next question? B. All right. Explain one importance of nucleic acid in cells. So this is very basic, and also you must not forget, why do we need nucleic acid? Okay. We need nucleic acid because this is the molecule which carries, is a molecule that makes up your DNA or RNA. For humans, we have DNA. Okay, for other organisms like virus and all that, they have RNA. So what is the importance of having nucleic acid molecules? Okay, so let's look at the most important fact that you have to remember. It is to carry genetic information atau membawa maklumat pewarisan. We call it hereditary information. That's why you have the DNA. DNA is the blueprint. It carries the information for your cells to make molecules like protein and so on. Okay, you have that information stored in the DNA. So that your DNA, the basic structure is actually the nucleotide. Okay, next, another point you can write is, because there are 2 marks, you have to write 2 points. 2 marks, you must have 2 points. Is to determine the characteristics in living organism. That means the kind of features that you have. Example, the color of your skin. the type of hair that you have, whether curly or straight. All of this information, all right, the features, is based on the type of amino acid that you will produce. And what kind of amino acid you will produce is actually determined by your DNA. Okay, so that's why everybody in this world is different, because they have different composition, all right, of your DNA, right? And therefore it makes different amino acid, and also you have different protein that gives you different features. All right, okay, so some people are dark skin, some people are lighter skin, and then some curly hair, ability to roll the tongue, not able to roll the tongue. All these are characteristics. Okay, so all this information is stored in your DNA. Okay, one more with an extra point, right? It contains the genetic code lah, all the codes for making protein, okay, or making the amino acid is stored in the nucleic acid. That's why you need the nucleic acid, because your protein is the basic block of life. Every living thing that we know of, scientists know of at the moment, everything that has life has protein. Okay, so your protein is the basic building block of life of cells. You need this code to make the protein, and this code is inside your nucleic acid, which is your DNA. All right, okay, so it's very simple. Again, this is we start with a question that is 6 marks only. That means the either question 1 or question 2 is 6 marks. Then question 3 and 4 will be 7 marks. I'm sure you know the format by now, yeah? Question 1, question 2 is 6 marks. Question 3, question 4, paper 2 is 7 marks. Question 5 and 6 is 8 marks. Then question 7 and 8 is 9 marks. All right. So later on, we go to question 9, 10, 11. That one will have more marks. Okay, that would be the section B lah, right? Okay, now we still have section A. Okay, any 2 points here, that will give you the maximum 2 marks. Okay, next one. So the question will go more and more difficult usually, all right? The first question is always the easiest. And as you notice, as you go down the question, towards the last question of the last bahagian, which is the C or whatever, sometimes they ask you to apply the principle in certain situation. They give a situation as application. Application is a higher level. It's more difficult to answer. Okay, so the first one is like just now the drawing, or that one is the easiest one. Okay, now exposure to harmful radiation causes changes in the original nucleotide sequence as shown in the diagram below. Okay, now I'll show you the diagram of an original sequence. So a normal cell with normal DNA should have this sequence, okay, where you have your nucleotide bases, T for thymine, A for adenine, TAA, C is for cytosine, T for thymine, G, CAGGT. These are the nitrogenous bases. We know there are 4 types of nitrogenous bases, okay, A, C, T, and G. Now this is the correct order. We call it the original one. Now let's say this person has been exposed to radiation, let's say gamma rays, okay, or higher doses of X-ray or any radiation that is harmful. All right, now it can cause the sequence to change. Like now you get this, okay, look at this sequence. This is a modified sequence. Now what you need to know is, you can see there's a slight difference. Okay, the slight difference here, the rest are the same except for this one. Wait, okay, question first. Explain the effect of nucleotide sequence change. Okay, now where's the change? Identify the change first. The change is here. You see here, okay? The TAAC is still the same. All right, then you have the, after that you have the GCA, GGT. It's the same, except for that particular nucleotide base, the thymine has been substituted with cytosine. Exchange already, changed already. It's no longer T, which is thymine. All right. Now, what kind of effect will this have on the production of protein? Making protein. Making protein is what do you call based on these 3 sequence. Okay, I'll show you. like TAA, for example, it is the codon. We call it the codon to make a particular amino acid. Now see the next one. Supposed to be CTG, right? Now what happened is now it becomes CCG. So this is the code for another amino acid. So instead of making the normal amino acid, because your code has changed, now your cell is making another type of amino acid, which is not normal. And this amino acid may not even function. So it causes your cells not to be able to produce the protein that your cell needs. Okay, so this is what you should answer. You must talk about the sequence has changed. So the amino acid that you produce is not the same anymore. All right, now 2 points. Where are you going to get your 2 points? Okay, let's look at the answer. So your points, you can talk about this type of mutation whereby only one nitrogenous base has altered. We call it point mutation or gene mutation. Okay, if you learn earlier, you notice there are 2 types of mutation. One is chromosomal mutation, the other one is gene mutation. So the point whereby your gene has been substituted, or this is substitution, all right, or it has been deleted or inserted, this is called point mutation. So this, you give the name mutation, gene mutation, you will get 1 mark. Okay. Now, so what does it incur? So you have this mutation of gene. What is actually happening there? Base substitution occurs. Instead of having that T, right, you have been changed to another one, C. So you change the base already. It's no longer going to be giving you the code to make the correct amino acid that your body needs. Okay, so this changes the genetic code, or we call the codon. The codon is got 3 codes, the ACT or TAG or GGT. All these 3 is actually a blueprint. We call the formula to make a specific amino acid. So because your code has changed already, the amino acid that you make is not going to be the original one. It's going to be another amino acid. And that causes your protein to be formed may not be functioning. All right, okay, so the P3 changes the type of amino acids that are synthesized. So the amino acid that you make is not the correct one. And when that is not the correct one, your protein that you make, because amino acid is a protein, right? So your protein that you make is the wrong one, and it cannot function. You do not have the, for example, you do not have the ability to, let's say, produce melanin, the brown color pigmentation of your skin. So you may be albino. Albino means white color, right? Your skin doesn't have that brown color, the color, right? So lastly, so you happen to synthesize a new protein, and this new protein may not be able to function, cannot function. So you have the penyakit, you have the genetic disease. Okay, just because one of the nitrogenous base has been altered, it causes the molecule that you make, the amino acid, is not the correct one. So you cause this kind of change in your, you cannot produce the protein that you want to produce. Okay, all right, so this is also 2 marks. This is question 2 actually in the format. This is easy questions, only 6 marks. So 2, 2, 2, this is a question C. And you just need to take any 2 points. Okay, any 2 points is always more points than you need. So write as many points that you can think of, because some of the points you may not get, and the other points may be able to cover back. Okay, so all these, you just choose any 2. All right, you write all also, very good. That means you really know your facts. Okay, now that's question 1, yeah? Okay, today our lesson will be very long, right? Because I know that very long lesson is very, very stressful, very tiring. So I only have 3 questions. All right, it's all about genetic engineering. Okay, next, it's all about genetics, right? Question 2 is a longer one. Okay, 8 marks. 8 marks is about question, what? 5, 6, 7. Oh, wait, 6, 7. Question 6 or question 7 in your format of exam. All right, okay. Diagram below shows a cell with an abnormal gene. Gene yang tidak normal. Okay, it's being treated by inserting gene X. So this is gene X, through a technique done to cure a disease. So this is a technique done to cure a certain disease. I hope you can remember what is the name of this technique. Okay, you have learned it in chapter 15, Form 5. One of the applications of genetic engineering is to cure disease. And the technique is known as, okay, the first question asks you, what is the name of this technique? Okay, is there anyone who would like to answer? Do you have the answer for this? All right. Ripa, I can see Ripa, you're here. All right. All right. You understand what I want to tell you. Good. So, do you have an answer for this? Do you know this technique? All right. If you can, just try. I know that many of you are already starting to do your revision because I'm sure your teachers have been finished teaching, right? Because it's almost like, 1 month more, one month plus more to your exam. And next month is your Ujian Amal Science, right? Okay, so what technique is this? Any idea? Normal gene? Okay. The technique is not called normal gene. That gene is normal gene, correct? Gene X is normal gene. So Ripa, you have already answered the gene X actually, yes. But what is the technique? Yes, Wing Han, correct. It's called gene therapy. Yeah. Gene therapy means, therapy actually means rawatan. Means is to, you want to make something better. You're using gene to correct the problem. Because the problem is this cell has a faulty gene, has a gene that is rosak, right? That you want to take out and you put in a good gene, the gene that is normal. So this technique is called gene therapy, or in BM we call therapy gene. This is one of the things that you learn in your chapter 15. Right? Chapter 15, you learn about therapy, gene therapy. You learn about DNA profiling. You also learn about the modification, GMO, all right, GMO and the modification of the gene so that you get a new organism. Right? There are many, many techniques there. Okay, so one of these is the gene therapy. And also producing insulin. Remember, producing insulin using bacteria. That you must not forget because that is a popular question. How do you engineer your bacteria to produce insulin so that you can make insulin for these diabetic patients to use? You can inject yourself as a diabetic patient. Okay, question 2. Name gene X. This one, Ripa, you already answered this now. The gene X is the normal gene because this is the gene that you want to put inside. You see the question diagram 1? There's a cross here. That means this is a faulty gene. Inside here, we don't want that gene. We want to put in, take out the faulty gene and put in the good gene. So this is actually the basis of genetic engineering. You are engineering, you are manipulating. You are trying to do something to the composition of the sequence of the DNA so that you will treat certain diseases. You can make the diseases better. Okay, now next question. State one disease that can be treated using this technique. What diseases in your textbook, right? In your what you have learned so far, what are the diseases that can be treated using gene therapy? Okay, anybody knows this answer? Okay. So Ripa, yeah, okay, understand. Good. So what is the disease? You have to remember every question or every topic here. What disease here that it can be treated using this method and so on. You need to remember 2 examples, just to be on the safe side. If you cannot remember 2, never mind, at least have 1. Okay? Then anytime the question calls for it, you can give the example. And even if you're writing for essay, if you have an example, it strengthens your essay because in the essay, sometimes we ask for points. One of the points is for you to give examples. Okay, next, dystrophy. Yeah, very good. Muscular dystrophy, yes. Cystic fibrosis, all right, muscular dystrophy, and cancer even. All right, all these can be treated using gene therapy. All right, this one is also in the textbook. I've seen this, yeah. So if you stick to the textbook, you can never go wrong. Remember, every time we want to make menggubal, means make the marking scheme for SPM question and so on. When you make the question, you're gonna make the answer scheme, right? They will always refer to your textbook first because the textbook is like the Bible. Okay, all the facts inside there should be correct. So if you follow the textbook, okay, this is diperlakukan oleh KPM. So the facts cannot be wrong. If they're wrong, the teacher maybe is an error. They already tell you lah, ERRATA, right, error. But then you follow it, then you should be quite safe. Okay, next question. More challenging. This one. This one you must, a lot of students must be thinking, "Ayo, what is this thing? I never learned before." Okay, active research has been ongoing in recent years using clustered regularly interspaced short palindromic repeats, or we call it CRISPR technology. You may not have heard it, but actually it sounds very complicated, sounds like very changgih, sophisticated. Actually, what is it about? It's actually the same thing as what we've been talking just now. It is actually replacing the faulty gene and putting the good gene inside, the normal gene inside. So it sounds very classy. The name is like so changgih, but the basic principle is very simple. Just take out the thing, you cut first, you identify what is wrong, you cut it not using scissors, using restriction enzyme. Whenever you want to take out a segment of DNA, okay, there are no scissors that are so small that you can go in and cut. We call it cut, but not really cutting using scissors. We cut using restriction enzymes. Okay, you take it out and then you put in the segment of DNA that has the correct sequence. Okay, and then you replace it. That is actually what it means by this term. Okay, just to show you. Question first. Based on the diagram above, explain the use of CRISPR technology in gene therapy. Okay, a lot of students don't know how to answer this because they think, oh, I have not seen this. Actually, the technology sounds very changgih. It's just very basic. You see the answer, you will smile because it's what you have learned before. Okay, before that, just tell you what it means. Sorry, excuse me. CRISPR allows, this technology allows scientists to modify DNA in living organisms. So basically, it's like this only. You take the unwanted sequence, the sequence yang rosak, okay, with the faulty gene, the gene that gives you the problem, all right? Take it out, then you insert, we call it splice, okay? Insert the sequence that has the correct sequence, the correct one, that no penyakit one, put it inside to replace it. So here, scissors, not real scissors, we use restriction enzyme atau enzim pembatasan. Okay, and then you see you delete delete that part that you don't want, right? And then now you insert the gene that is normal. All right, okay, so let's see some comments here. Yeah, the video will be saved here, Ricky. Yes, you can click back the same link. Later, you will be able to see again. Okay? Yeah, good. Identify the abnormal genes first, correct? You need to know what is the problem you want to treat, right? Okay, so what is your, only 2 marks, very short only, this answer. You need only 2 points. Okay, first, you got to, Ripa, is it Ripa who answered that? Yeah, Ripa, you have identified the abnormal genes, correct? Huh? You must identify what you want to take out. Okay, the abnormal gene is identified. P1, first point. P2, the abnormal gene is cut. You can add on to say by using restriction enzyme. You don't say using scissors. You say using scissors, the whole idea P2 wrong. If you don't say, it's still okay. But if you say the wrong thing, then the whole sentence is wrong. Okay, gene, we call it abnormal, gene abnormal dipotong, which we remove. Then you got to put in normal gene or the gene X is inserted or added to replace the abnormal gene. Okay, this is the principle. You need to explain the principle only. Don't need to talk about how it's actually done in the lab. All right, it's not for you to know. It's too complicated, too technical. Even I don't know. All right, so P4. So now the new gene has been inserted. This cell with the new gene will be able to function normally because it has the correct sequence already. It has the correct DNA. All right, it has the new modified DNA sequence. All you need is 2 points from here. Okay, because they just want 2 points. Always remember, before you answer, look at the marks. Okay, 2 points means you need, 2 marks means 2 points. If you have 3 marks, please make sure you have 3 things to say. Otherwise, you lose out your points. Okay, next, now you go to question 3. Question 2, question 3 goes to some more. Okay. Right. So this is application. Application. So you see the question, from basic answering things that you know, simple things, remembering only. Now we go to application. Okay, the diagram below shows the result of a DNA test conducted on a skin sample that was found at the scene of a crime. So a crime has been committed. Okay, so the skin sample probably the robber or whatever was scratched, right? There's a tussle, there was a pergulutan, right? The person was hurt, right? It could be blood, it could be skin, any or hair. So what happens at the scene of the crime? They have to take evidence, right? So inside the cell, any cell will have nucleus. Any nucleus will have chromosome. And from the chromosome, there will be DNA. So as long as it has cell, you can do DNA tests. As long as it has cell, right? So DNA was extracted from the sample and compared with DNA from 3 suspects. So happened there are 3 people who were suspected could be the criminal, the one who committed the crime. All right, so I want to see which skin sample, right, that was collected there belong to which suspect. Okay, so where's the question? Question here is, based on the diagram given, who is most likely the person who committed the crime? Okay, so who do you think? Are you able to know how to see this? Okay, in the textbook, why we use virus, not bacteria? Okay, Ripa, you have a question there. I will address your question now. We use virus because the virus that we use, not all viruses are harmful. All right, because there's a virus called adenovirus. Adenovirus, it does not carry any disease. What we need to do is we use that virus to carry, because the virus will, we put the DNA RNA into the DNA into the virus, let the virus infect the cell, and then the gene will go inside. We make use of the virus to infect. Bacteria, on the other hand, sometimes you can use certain bacteria for certain job to do. You cannot be using the same bacteria for all the jobs, okay, all the tasks that you need to do. For example, if you want to make insulin, you use bacteria, yes, E. coli, Escherichia coli, right? If for certain gene therapy, normally we use adenovirus. So not all bacteria is harmful. Same thing, not all virus is harmful. Okay, so we use bacteria and virus to carry the genetic information to put it into the cell. Yes, both your answers correct, Ripa and Wei Han. Okay, I think you know how to see. Very simple. You take a ruler and then you line it up with, let me draw. I'm going to draw the line here. So you line up with, you imagine your ruler, you draw a line here. This is the thing you want to see. You see, and then you put it against which one? Okay, it fits here. So put a tick here. Find the one that has the most tick. All right, so the second band here, the pattern here, it fits here. Okay, this one boleh, this one got, this one also got. Okay. Then this one, take a ruler and then you go down the line. This one tiga pun ada. 1, 2, and 3. Right? You see which one matches the most. Then this one, this one got, right? This one don't have. This one got. All right, and then I'm going to go down. All right, this one don't have, and you go to the next one. Here. So this one and this one. You see which one has the most matches of the band. Band is all these lines that you see. All the lines that you see on the DNA. We call it, this is a DNA fingerprint. We call it a fingerprint because fingerprint is unique. Not the fingerprint on your finger. This is called DNA fingerprinting is called the picture. The picture of the DNA after undergoing the test, all right, which is called the DNA profiling. So you find that this one is the paling banyak, most matches. So usually most matches means highly likely this skin sample belongs to this person. Okay, suspect 2. All right, you're able to answer, but how to explain? Next question, explaining it. All right, explain your answer in C1. So this one 2 marks. All right, 2 marks, yeah. All right, so let's see how to, just need 2 points only. I'll give you more. Now the DNA pattern, we call the bands, all these black lines, you see the DNA pattern, or we call the DNA fingerprint of the skin sample and suspect 2 are the same. It may not be exactly the same. You can also say very similar. So happen this is exactly the same, all right? If you don't say the same, it's okay. You say very similar. So the most matches, most like up to 90-something%, right? That means very likely is that person, up to 95% and above. Okay. Now everyone has their own set of DNA. Every DNA of a person is different. That's why we look different. Okay, we are humans, all of the same species, but our combination, all right, of the genes, in the DNA there, the nucleotide, the codes there are different. So everybody's individual DNA is unique. That's why there are no two similar people, except if you are identical twins. Because if you're identical twins, your genes will be similar, exactly the same, because you come from one sperm and one ovum. And they just happen to split into two individual only. The composition of this twin number one and twin number two, exactly the same. Okay, but we are all not related, right? Definitely DNA is different. So everybody have their own pattern, the DNA. So the technique used here is called DNA profiling, right? They give you a mark for saying the name lah, right? So the most important thing is you must say the pattern, the bands on this DNA fingerprint is very similar or matches or has the most matches with the suspect number two, has the most matches. Okay, right? Clear? Okay, next. Question 3. Question 3 is an essay question. Same code DNA lah. You see the matching the matches, Ripa. You see the pattern matches most matches. Okay, that means paling similar. All right, or they say look so happen to be the same. Okay, this is essay question. All right, essay question means more things to write. Okay, and a little bit tougher lah. Okay, now the diagram below shows a pair of homologous chromosomes. I hope you remember what is homologous chromosome. A characteristic is determined by a pair, right? Any characteristic, skin color, ability to roll the tongue, whether you're tall or you're dwarf, right? Whether the color of your eye, whether it's blue or whether it's brown or black, is determined by a pair. Always a pair, because one is donated by the father, one by the mother. You will have a pair of alleles. Okay. Now, example here is given capital R and small r. They represent the alleles for the ability to roll the tongue. So they give you an example, ability to roll the tongue, right? So here, this shows homologous chromosome, pasangan chromosome homolog. They are found on different locus, on the same locus but on different chromosome, same place but on another chromosome. We call it a homolog. Okay, so this is found here, and the other one on the same place, locus, but on their pair. The pair is called the homologous chromosome. Pasangan dia. All right. So one maybe father donated this, then the other one donated by the mother. All right. So you always have the chromosomes in pairs. That's why your number of chromosomes always is nombor genap. It's always even numbers. You don't have odd number chromosomes. Okay. Explain how the characteristic of ability to roll the tongue is determined. How does having this allele determine whether you can roll the tongue or you cannot roll the tongue. Okay, this one go back to the basic, no? The first lesson when you learn about the Mendel, right? The first chapter, the first lesson on 11.1, all right? The monohybrid crossing, we first learn about dominant allele and recessive allele, all right? Yeah, the dominant and recessive. So we have two types of allele, right? Dominant and recessive. All right, so capital R always represents the dominant allele. Dominant means stronger, right? But don't use the word stronger. You must use the word dominant. All right, dominant allele will show. We call it will manifest. Will show the feature if there is at least one. You need at least one dominant. The other one, it can be dominant, it can be recessive, doesn't matter. As long as you got one dominant, it will show that feature. Okay, or it show the trait. And small r will represent the recessive allele. Dominance will happen, scientists have found that able to roll the tongue. Dominant is the ability to roll the tongue. And recessive one controls the ability to not able to roll the tongue. Okay, so both alleles are located at the same locus. You can see at the same place, but on a different chromosome or the different pair. We call it the homolog. is on a different homolog. Okay, the same pair. So a pair of heterozygous, that means if it's capital R and small r, one dominant and one recessive is called heterozygous. Hetero means not the same, right? One dominant, one recessive. It will have the ability to roll the tongue because it follows the dominant. Dominant, like we say, is stronger. As long as you got one, it will show the ability to roll the tongue. Okay? If it's recessive, one doesn't matter. One is not strong enough to show. You must have two recessive, then only can show the not able to roll the tongue. Okay, this one is revision a little bit of your Form 5 book, chapter 11.1. Okay, then if you have two dominant, we call it homozygous allele. You have two, is capital L, capital R, always a pair one. It shows able to roll the tongue. Because one dominant is already able, two also definitely able. Okay, and then what about another case? If you have two small r's, which is homozygous recessive, recessive homozygous, two small r here, then only you have the inability to roll the tongue, unable to roll the tongue. So you have to explain all these, the effect of the dominant and the recessive. Then only it shows you how it determines whether you can roll the tongue or you cannot roll the tongue. What is the condition? You roll the tongue either your capital R, or capital R, small r. If you have small r, you cannot roll the tongue. Okay, so you need any 2 points from here. All right, here, all right. Yes, Weihan, correct. So allele, capital R is dominant allele. Yeah, allele, small r is recessive allele. Yeah, that's how you, if you write that one, yeah, thank Weihan, you can get P1, right? But only 1 point because you didn't write anything else. So you want to get full marks, you must write some more. So what you wrote there just now is enough to get P1. Okay, right. Now, next question. Okay, let's go on. 20 marks. So diagram B. So this one, a little bit of application and ask me to explain. You know about rhesus factor, right? This is the type of antigen, a type of antigen found in red blood cell, on the surface of red blood cell. Either you have it or you don't have this antigen. So rhesus factor is controlled by a pair of alleles. Okay, if you have Rh plus, the Rh plus is considered dominant, and then the Rh negative is considered recessive allele. All right, okay, Rh dominant, Rh positive is dominant, recessive is a negative. Right, look at this scenario. All right, they show scenario given to you. Father is positive. All right, that means he's rhesus positive, but it doesn't matter whether it's dominant or dominant recessive, as long as dominant. Mother is recessive. Okay, then they can have children, right? They have children, they have 3 children. First child is rhesus positive, second child is also rhesus positive, and the third child is happen to be rhesus negative. Now the rhesus negative is because the father will also have one rhesus negative. That means the father is not homozygous. The father is one positive allele, and the father also has a recessive allele. That's why this baby is actually Rh negative, Rh negative. One of it comes from the father. You see, always contributed one by the father, and this one contributed by the mother. So that's why this child can be rhesus negative, because got one from the father here, and the other one definitely from the mother. The mother is rhesus negative, definitely is Rh negative, Rh negative. Okay, so the other one comes from the mother. So these two made this baby. All right. See that? So this is the child. All right, where's the question? Question is here. Based on the diagram here, draw the schematic diagram of inheritance of rhesus factor in the family. So because you have a child that is rhesus negative, that's how you know the father cannot be Rh positive, Rh positive. Because if the father is Rh positive, let's say this one happened to be positive, let's say this one happened to be positive, This happen to be positive. That means this one also will be positive, right? Not. You must get one from the father, get one from the mother one. That's how you can make a baby. So if this is positive, that means this baby must definitely, because positive is dominant, this baby is definitely positive. Okay, but because the fact the baby here is negative, that means the father must carry one negative allele there, the recessive allele. Okay, how to draw? How many marks usually given for this kind of diagram? Up to 7 marks, from 5 to 7, depends on the question. So look carefully, 5 marks, you just need minimum of 5 points there. All right. The father is carrier. Yes, carrier of the recessive allele. Correct. Zhe Qian is a carrier. Carrier means it has, I wouldn't say it carry the disease. It's not carrier the disease, but it has one recessive allele. Yeah, that's why the baby can have negative, because one of it came from the father, the negative came from the father. The other one sure from mother, because mother is negative. If negative, sure negative. Okay, you can analyze the situation. Now, how to draw? Okay, now any 5 points, let me tell you how they give you marks. They must look at the diagram secara horizontal. You know horizontal? That means from left to right, 1 line, 1 mark. Another line, 1 mark. We don't look from top down. We don't look from top down. We look from left to right. Okay, so like this, you must write the parent first, of course, father, mother, right? Now, this one, you must look at the genotype. The most important part of this question is to get the father genotype correct and the mother genotype correct. Because if the father genotype and the mother genotype is wrong, definitely your entire question, your answer is all wrong indeed. Because the 2 important thing that is wrong, all your answer all wrong indeed. Okay, so that's why I told you how to analyze it just now. If the baby is negative, that means one must be from the father, one must be from the mother. All right, so the father is like this, the mother must be like this, and this one, the whole row here, 1 mark. So you look at it this way. This entire here is 1 mark. See that? You must write here also. If you don't have the word genotype, you draw. A lot of students very lazy, right? They want to take a shortcut, right? They just write RH positive, RH negative, and you just draw. Left hand side, nothing. No genotype no meiosis no fertilization nothing. Only like this, definitely cannot, because it will penalize you. They will penalize you. So make sure it's row by row, complete. This one correct, this one correct. All 3 things other, then we give you 1 mark. Okay, now we carry on the next row. All right, next one. Meiosis. Don't forget to write meiosis, because Mendel's first law of segregation. They say that law of segregation says that during formation of meiosis, every gamete will take 1 allele only. There's no such thing gamete can take 2 allele. So it must split. Okay, gamete must take 1 allele, then the other gamete will take the other allele. So you get 1 mark here, just for the 2 lines there. All right, the 4 lines. And then the next row, gamete formation. What kind of gamete will be produced by the father? All right, so around 50% of the sperm of the father will be carrying the Rhesus positive allele. This is Rhesus positive, in case you cannot see it. This is Rhesus positive. Then this is negative, right? So 50% of the sperms will be carrying Rhesus negative. And then for the mother, because the mother all also the same, positive, so definitely is negative. This whole row here, gamete, and all these 4 circles, and this will be 1 mark. Okay? Any questions? I think so far. All right, next one. Next row, fertilization. Okay, how do you get a baby? There must be fertilization, right? From 1 sperm from the father and 1 ovum from the mother. So you take 1 sperm from the father, 1 ovum from the mother, and combine. Okay, so the best way to combine is, I always tell my students, 1 3 1 4 2 3 2 4. You will never go wrong. I will see students sometimes simply draw the line, because they don't understand how to get this line. So the correct way is, you just remember, you just consider this as first number 1, number 2, number 3, number 4. You must take one sperm and combine with each of the ovum. Okay, so 1, 3, you get this. Then 1, 4, you get this. Then 2, 3, you get this. 2, 4, you must use up all the possibilities. Okay, 1, 3, 1, 4. 2, 3, 2, 4. Cannot be repeated. Cannot be 1, 3 and then 1, 3 again. Cannot be 2, 4, 2, 4 again. You don't have your 1, 4 and 2, 3. Sana. Okay, now this is the best way to remember 1, 3, 1, 4, 2, 3, 2, 4. Then you fill up. Okay, I get this one. RH positive, negative here. 1 dominant, 1 recessive. 1 dominant, 1 recessive. And this one is both also recessive. Okay, this line here, fertilization line will mark. And the next one, genotype of the offspring, that means anak, right? What kind of allele will the baby have? So you write all that one, also another mark. You see here maximum maybe. In case you any part salah, you still got marks at the bottom for you. So phenotype, what does positive, negative mean? It means it is rhesus positive, right? RH positive, negative, also rhesus positive, because positive is dominant. Then the other one is RH negative, definitely negative because there's no positive there. All right, so you must write down there the phenotype. What does it mean? What is the feature? And lastly, put in the ratio as well, just in case. Even though they don't ask you, what is the ratio of rhesus positive to rhesus negative. Never mind, they didn't ask you that, you just put in also. So you find that 1 to 1 or 50% chance. All right, every time the baby is born to this father and mother, there will be a 50% chance of whether it's a positive rhesus or a negative rhesus baby. Okay, so here, you see here, 7 marks here. 7 marks. Back to the slide. See, 1, 2, 3, 4, 5, 6, 7. Then we just choose any one. We always menguntungkan you one. So make sure you label nicely. If you don't have this one wrong, nothing, not to say wrong, you never write this, you'll be penalized. You don't get 5. Definitely you will get only 2 maybe, or 1 or 2 or 3, even though everything is correct. Okay, please don't be lazy. You must write this. Okay, now next one. Continuation from the question. The first and the second child are healthy. Right? They are normal, no problem. But the third child, this is just baby, right? Died shortly after birth. Now, this one the question get a little bit more difficult, right? Because it's application of what you really learned. So based on your knowledge about rhesus factor, explain why the third child died, cannot survive. Okay, the first and the second one boleh, right? No problem. But the third child, see, is rhesus negative. You can see the difference here. Rhesus negative, right? The second child is positive. The first child is also positive. So positive, no problem. If the next child is a negative, right? The mother is negative, all right? Now, what happened here is you have to know about the antigen-antibody reaction there, okay? 3 marks only, not a lot. All right, so let's look at what happened now. Now, when the first baby is born, or the second baby is born, first baby, second baby, no difference. Because the baby, right, wait, let me go back to the picture, right? Because this baby is positive, right? Positive, that means got we call the antigen there, right? So once the mother gives birth to the first baby, there will be blood breaking, blood vessels breaking. So this antigen, the red blood cell of the baby, will enter the bloodstream of the mother. Enter the bloodstream of the mother, and this antigen will cause the mother to produce antibody. Okay, it will go inside the mother's blood, right, through the umbilical cord, right, because the placenta is broken, right, because when during childbirth, it's very, very stressful, right, the blood vessels will be breaking. So there will be a moment whereby the antigen of this rhesus positive, right, antigen will enter the bloodstream of the mother, and now the baby is removed, the baby is already born. The antigen in the mother's blood is now stimulating the mother to make antibody after the baby is born, right? So the first brother or the first and second child, after born, the antigen of that rhesus possibly go inside, and now it stimulate the mother's blood to make antibody. Okay, now, so what happens is the antibody of the mother is being made inside the body, right? The antibody will now become more and more, right, during the pregnancy, the next baby. So got time, the time to build up, right? Build up the antibody. So now the antibody can go inside the bloodstream and go and destroy the cells of the fetus, the third one, the not second pregnancy, the third one. Actually, the next baby, the second pregnancy, not second pregnancy, sorry, the third one, the third baby. The third baby there, because it has got, now the antibody go into the baby, is going to destroy the red blood cells. Okay, and we call this erythroblastosis fetalis, the medical term, whereby the red blood cells, they will clump together. They might even burst, break open, destroy. So the fetus that is born, right, the baby is born, will have complications. Okay, that's why the baby, the next one that is born, will have complication. All right, it cannot survive. So it can also cause mental retardation of the fetus. All right, so you have to explain in that term lah. The most important thing to talk about is the formation of antibody. Okay, put different color here, red color. Formation of anti-D antibodies. All right, because of the antigen, the baby is positive. The baby is positive, but the mother is negative. That will be a problem. All right. The first pregnancy, no problem. All right. Because once the antigen go into the mother's blood, the baby is already removed. The antibody does not go into the blood of the baby. But the next pregnancy can have problem. All right. The antibody can go inside and attack the blood or attack the fetus, that it destroys the red blood cells. Okay, right? So any 3 points from here. All right. So how can antibody go to, actually, because it's also positive, blood clotting in the fetus. Wait, Irish when you attack the third child. Yes, correct, Weihan, correct. That's what you meant now. Because the third child, once the baby is removed, the doctor cut the umbilical cord one, isn't it? Cut the umbilical cord, the antibody doesn't go in. But the third baby, during pregnancy itself, will have complication already. Because when the mother is still carrying the baby, the antibody can go in. Okay, antibody can go in and attack the red blood cells. Okay, now the problem is because the third one, the first one and the second one, actually it shouldn't be two. Yeah, it should be the second one got problem. Yeah, not the third one. Yes, correct. So Jehan, the question there is, the question they shouldn't, because in our textbook, we only say the first and the second baby, right? Antibody correct, maybe possible. It should not be saying the third baby. Actually, the second baby got problem, right? The second baby. So you follow the textbook, right? I don't know why the question, I didn't, really modify the question. I just used it as is. So good question there. All right, any 3 points there? All right, okay, the next part of the question here is, last part. Karyotype, this is the pattern of the normal human. So normal human, you should know how it looks like. How many chromosomes? There should be 46, all right? And you can see XY here. It should be a male, right? It's a normal human, a boy. Now there are 2 types of human chromosomes. You can see there are 2 types here. You should know what I'm talking about. State the differences between the 2 types of chromosome. Okay, who can tell me what are the 2 types of chromosome here? There are 2 groups of chromosome here. Anybody? Chen or Wei Han or Ripa? I think only 3 of you here. I don't know about the rest. Actually got 20 people here. Okay. So blood clotting in the fetus, that's correct, and also the blood clotting, agglutination. So sex chromosome, yes. What's the other one, Chen? Besides the next group, one is sex chromosome, which is X and Y. Then the others are known as what? The others are known as, yes, correct, autosome and sex chromosome. Okay, I've got very good students here, right? Where you are very good, right? You know your facts. So the two types of chromosome here are the autosome and sex chromosome. So what they want you to do is to tell the differences, state the differences. So when the differences, you don't need to talk about similarity already. Look carefully, read the question. They only want differences. So that means perbezaan. No need to talk about differences. And so no need to talk about similarity. Okay, the best way to write differences is you're comparing, right? You're comparing. So please make sure you compare point by point of both sides. All right, don't make the mistake of writing everything about autosome in one paragraph, and then after that, write everything about sex chromosome in another paragraph. That's a wrong way of comparing because we cannot see the differences. Okay, you must always compare sentence by sentence, and then only you get 1 mark like that. Not talk about autosome first, finish the story, and then talk about sex chromosome. Wrong. All right, you must talk about this. So, for example, one group of chromosome is known as autosome, while the other is known as sex chromosome. Okay, give the name lah, because the name is not given, right? You get 1 mark. Okay, next, talk about how many, right? There are numbered. Autosomes are numbered from number 1 to 22, and they are in pairs. Okay, sex chromosomes are numbered the 23rd pair. The 23rd pair, it's considered as X and Y. It could be XX, it could be XY. Okay, depending on the gender lah. Next one, autosomes control all characteristics of somatic cells. That means all the cells inside your body that are not you, they are not gametes. Okay, not the sperm, not the ovum, they are considered as somatic cells. Like your skin cell, your cheek cells, your hair cells, liver cells, all of them are somatic cells. Okay, and then for sex chromosome, they control, the function is to control the determination of gender, whether boy or girl is based on the sex chromosomes. Okay, remember to put it one sentence, compare in one sentence. Okay, don't finish one paragraph by paragraph. Okay, any two points. Okay, last question. I think the last question. Yes. Now, diagram 3.1 and 3.2 shows the character of two persons with genetic diseases. Okay, now you got to compare. All right, they look almost similar. Everything you got to check properly. Everything looks like everything is in sets, right? We look for any abnormality in the number and the type of chromosome. Okay, you will see there is difference here, right? Compare and contrast the two genetic diseases shown. Can anyone tell me what is the genetic disease for 3.1? And what is the genetic disease for 3.2? Anybody knows? I think you're good enough to know. I think you can answer lah. Ripa, Zheqian, and Weihan. I think you all know. You should have the fact at the back of at the tip of your fingertips, because all these are things that you should know already. Turner syndrome. Okay, for which one, Ripa? This is a very dangerous thing, because if you just write like that, if we are to mark your paper lah, we don't know which you're talking about. Are you talking about 3.1 or 3.2? You have to mention which is which. Okay, so which one? Ripa. Turner is which one? The one on the left, is it? So you always assume the first one is the one on the left, right? Turner. Yes, always write 3.1. Okay, the diagram shown 3.1 is Turner syndrome. Yes. Yeah, Rainy. That's the correct of writing it. You must put 3.1 Turner syndrome, 3.2 Klinefelter syndrome. Okay, the syndrome is wrong spelling for the second one. Okay, now let's look at what can you compare. Compare and contrast. Please don't forget the similarity. A lot of students just go straight to comparing the differences, and that's where you lose marks. Here, 8 marks. Okay, how do we give you 8 marks? Okay, look, give similarity. Similarity, you can just write both. Both are examples of chromosomal mutation. All these two diseases are caused because of a mutation called chromosomal mutation, because the number of chromosomes is wrong, or the type of chromosome is wrong. Okay, chromosomal mutation, not gene mutation, chromosomal mutation. Then both are caused by mutation. Maybe the person or has been, you know, the parent of this person, the parent of these two problems, the parent has been subjected to mutation. Mutation, X-rays, gamma rays, that causes the sperm to be lost, right? And then mutation occurred. Next, both are caused by non-disjunction. This is a fact that you must remember. All the syndrome that you learned so far, Down syndrome, Klinefelter, all this one, and Turner syndrome, are caused by what we call non-disjunction. Okay, the chromosomes do not split evenly during gamete formation. You got extra one, go to one side. That one you read that now. You don't have time to go through all this, but remember non-disjunction is important. That causes all these gametes that do not have the proper number. Okay, both involve problems with the sex chromosomes. So both problems are with the sex chromosome. That is the problem there. So any two, minimum you must write two of similarity to be safe. Okay, then some more. Both have fertility issues. Both the Klinefelter and this Turner syndrome, they have problem with producing the next generation. They have masalah kesuburan. Okay, now differences. Differences you can put in table. Just now the one similarity is put both in one sentence. This one you can put in table, but please write in full sentence. Okay, you can write in sentence as well without table, but if you want table, easier for the examiner to see your marks, to see your points. But make sure they are in full sentence. Try to write in full sentence. Okay, diagram 3.1 shows Turner syndrome. Diagram 3.2 shows Klinefelter syndrome. Okay, the one on the left is 3.1. It occurs in females, while for diagram 3.2, this condition occurs in males. Okay, that's one differences. Another differences caused by what? What is the problem there? Turner syndrome lacking one X sex chromosome. Klinefelter is caused by an extra X sex chromosome. Okay, extra one. Okay, next one, some more differences. 8 marks, right? You got 2 over there, you must have 6 over here. So total chromosome is 45 for Turner. This one total chromosome is 47. Compare in terms of this thing, must be both sex matching. Has underdeveloped or undeveloped ovary. That's why they have these fertility issues. They are mandul, they're sterile. They cannot have the next generation. For Klinefelter, they can have children, but they probably have problems because they have small testes. All right, production of sperm is not as well or as good as the normal person. Okay, has web neck. The neck has got folds here. All right, low IQ. Then this one long, for Klinefelter, long legs and arms, possesses feminine features like kewanitaan, all right? And you may have breasts also. So any feature, just give one feature. P6 is for any feature of Turner syndrome and Klinefelter syndrome. All right, now how to get full marks here? Now you must have minimum of 2F and then the rest must be P. That means if you write 2 similarities, then you must have 6 differences. All right, and then if you want to write the opposite, differences must have 2, and then the rest is similarity must have 6. But very unlikely for you to write to get so many similarity points. Normally don't have so many. Similarity normally is always less than your differences. So write your differences more. Okay, so that's how you can get your 8 marks. So make sure to be safe, have 2 similarities every time. You can write more, and then the examiner will choose for you, right? Which is correct. All right, okay. So I think that's about it for today. Right, it's been already 1 hour. Didn't know about it. So at the moment, this is the class I have for you now. In fact, you can check out my channel. I have many other videos that I've done before since the MCO. I've got a lot of videos there. If you find any questions or any topic that you would like to go through again, right, for revision, just go to my channel. I will share the link here. Right, let me share here. This is my copy the link here. Okay, and then here. Okay, this is my channel name. And then you can also PM me if you would like to ask questions. All right, I'll be more than happy to answer your questions. Okay, this is my Telegram name. Okay, so you can contact me lah there. All right. Okay, so Monday I will have a similar class, but just that I'll be speaking in DM lah, right? The same thing. So wherever you get the link, you'll also see the same thing. You just click on the same link, you will see the same video again. All right. I mean, for all my videos, it's always on my channel. You can always get it and see again. Okay, please share to your friends if you feel that your friends also need to see this. Yeah. All right. Okay, thank you very much. So I will see you hopefully another time, maybe 2 weeks from now, right? I need time to make my slides. Okay, all right. Thank you. Bye-bye. See you. If any questions, please PM me or Telegram me. Bye-bye.